Thank you. Good afternoon, and welcome to this inaugural Dean's Distinguished Lecture. I'm truly delighted that Dr. Michael Chang is here with us today to start off this new College of Medicine lecture series. As we all know, biomedical science research has become a team sport and requires different subject matter and technical expertise, including cell biology, molecular biology, systems biology, immunology, all multiomics and high-end imaging, AI, machine learning, and more. Given this reality, it's highly essential to expose our junior faculty members, students, and residents, and fellows to lectures and seminars on research topics that integrate efforts from multidisciplinary teams that have worked and are working together to solve major scientific problems. Therefore, the main purpose of this new Dean's Distinguished Lecture Series is to engage our College of Medicine research community with leading-edge science that involves both the depth and the breadth of scientific knowledge and technical expertise required to execute the project. This series will host scientists who are leaders in important areas of contemporary scientific endeavors and who have made seminal contributions. And also, we believe that this lecture series will lead us to develop further insights into building successful scientific teams and new ideas about diversifying and expanding our research programs at all levels. It's an absolute pleasure for me to introduce Dr. Rosenblatt, Executive Dean of the University of Illinois College of Medicine and Professor of Ophthalmology and Visual Sciences, and of course, Young Chair of Ocular Research. Dean Rosenblatt.
Thank you so much, Dr. Prabhakar. Thank you for your work in organizing this series. We are very fortunate to work in a community of scientists and scholars that are truly excellent. And that's really the best part of my job, is getting to serve you, to work with people who really are quite a bit brighter than I am. And what can I do as Dean to try to further enrich our community? As we emerge from this pandemic, and of course we haven't declared victory over COVID yet, what can we do to come together and to become better through bringing excellence here? And that's why we want distinguished lecturers to come forward. One, we'll have a chance to hear from Dr. Chiang. We'll also have a chance to perhaps chat with him a bit, answer some questions here, and also during the reception to follow. But also brings us together after a time we haven't had a chance to come together for so long. There will be a reception for those who can be here in person in the Alumni Lounge to follow, and there'll be discussion, there'll be some beer, there'll be some wine and some food. So that's a great thing. So I can't think of anyone more appropriate to come for our very first distinguished lecturer as Dr. Chiang. I've known him for almost a couple decades now, and it became very clear in the earliest parts of his career that he is one of the clearest and most innovative thinkers around. Of course, he's a pediatric ophthalmologist, already doing great work, but his skill set and his knowledge goes well beyond that, and we're so grateful that the National Eye Institute selected him as their director. He's only the third director of the NEI. The other two directors lasted at least roughly a couple decades apiece, maybe even longer. So we expect that we'll have Mike as the director of the NEI for quite some time. And again, his work isn't just in ophthalmology. He's leading trans NIH initiatives around data science, and I think we're all the better for that. So thank you, Mike. I'd also like to thank Dr. Paul Chan. One of the things we're going to try to do is, as we continue to this series, is to partner with departments, with faculty who want to bring eminent scholars, and Dr. Chan was kind enough to co-sponsor this event with Dr. Chang. And so Paul is the Panton Professor of Ophthalmology and head of the Department of Ophthalmology and Visual Sciences at the Illinois Ear Infirmary. And so Paul, you can more formally introduce Michael. Thank you.
Thank you, Dean Rosenblatt. It's a great pleasure for me and really an honor for me to formally introduce Dr. Michael Chang as our lecturer today. I'm going to be very formal and not call him Mike. I'll say Dr. Chang is the director of the National Eye Institute, as well as Mark I've known Dr. Chang for almost 2 decades. And just to give you some formal background, he's a pediatric ophthalmologist. He's board certified in ophthalmology as well as clinical informatics. His research, and he'll talk about this, it's at the intersection between biomedical informatics and clinical ophthalmology. He's known for his expertise in retinopathy of prematurity, telemedicine, informatics, EHR, and also genotype-phenotype correlation. He is also an adjunct investigator at the National Library of Medicine. He's published over 250 papers, and he's developed an AI system, and I've been fortunate enough to work with him in that space that received breakthrough status for the diagnosis of retinopathy of prematurity. Dr. Chang's been the director of the NEI since November 2020. Amazing to me how much gray hair you have since that time, because his hair used to be all black. So through the pandemic and through his new position, he's also co-chair of a number of working groups that go across the NIH. And this is what I think is so impressive about Dr. Chang, is that he's collaborative. He leads so many different spaces, and he doesn't just focus on vision science. So he's the co-chair of a trans-NIH working group for high-value data asset sustainability, chair of a trans-NIH clinical trials infrastructure working group, co-chair of a trans-NIH medical imaging working group, co-chair of the NIH AIM Ahead Advisory Committee, and co-chair of the NIH Common Fund Bridge to AI program. As if you don't have enough to do, Dr. Chang. But just to give you some background about where he's from, he was born and raised in Detroit. We had a conversation around flyover country, and flyover country apparently now is in, so it's great to be in the great city of Chicago. Went on to undergraduate at Stanford, medical school at Harvard, residency at Wilmer, Johns Hopkins, also fellowship. I first met Dr. Chang in New York as a young attending at Cornell, where he was under a K at Columbia, and then his career just took off. His most recent position prior to the NIH was at the KCI Institute, Oregon Health Science University, where he was vice chair of research and led many programs. He was the PI for a T32 program there, the K-12. He's a great educator, great mentor, led so many initiatives within ophthalmology and biomedical informatics. So, on a personal note, you're a tremendous friend, and thank you so much for being here to give our first lecture. Dr. Chang.
Thank you very much. I'm going to use your first name and say Paul and Mark, and Prab, thank you very much. I just want to emphasize that I feel like we're among friends here, and this is a real privilege for me. I think that Mark, you talked about bringing excellence in. I would flip that around and say, one of the things I love doing is visiting places that I think are doing excellent work. And you love to see friends sort of do kind of amazing things. And one thing I love about this atmosphere is that I think it's doing such good work in a really collaborative atmosphere. I used to say that, when I was trying to find people to work with, or we try to hire people, you wanted to find people who were nice, bright, and hardworking. And that 2 out of the 3 I felt like was never good enough. And I love that about this place, that the people, I feel like everybody that I know kind of fits that characteristic. And so I just wanted to show a couple, as both Paul and Mark had mentioned, we've known each other for a long time. And in 2011, there was a meeting in Australia. And we flew out there. We happened to be on the same airplane ride. And, this happened to be some pictures that I captured of my 2 daughters, with Mark and with Paul, the way that they looked back in 2011. And as I looked at this, I thought Mark looked about the same, but I'm not sure here. And this one was when they were checking in. One of my daughters really has taken a liking to both of them. And it looked like she was trying to say something to Mark. So with that, what I was asked to do is to say something about a little bit of the work that I've done and a little bit of the work for where I see the field going in a way that crosses disciplinary boundaries. And so I decided to call this artificial intelligence in medicine, essentially what we can learn about ophthalmology, about where we're heading as a community overall. Okay, so I'm going to try to do that. Just one last issue is that I've been in this position, as Paul said, at the National Eye Institute for about 1.5 years. And I've discovered that one of the things that we really have to do is communicate what we're doing with the people who are actually doing the work. And we've been using social media to try to do that. NEI director is the handle, and it feels ridiculous to always be trolling for followers. But the reason that I have been saying this is I used to always hate it when I couldn't apply for grant opportunities because I didn't know about them until right before the deadline. And so we try to socialize things like grants, other news that's of interest to the vision or scientific communities. So with that, let me go ahead and get started. First slide is what's the National Eye Institute? We were founded in 1968 by Congress and then President Lyndon Johnson as the branch of the NIH to manage national efforts in vision science. And what we do is we direct and fund vision research like every other institute at NIH. And one of the reasons we exist is that there's an impact of vision on quality of life. Multiple surveys have shown that blindness is one of those things that Americans fear the most. And but from a different perspective, there's enormous and broad impact of vision work on science overall. And this is something that we talked about over the past couple of days, that it's Nobel season right now. And one of the things that we're really proud of is having funded the work of 8 Nobel Prize winners. And the point that I want to make from this is that there's a lot of seminal innovations that have occurred in the eye because it's so accessible. If we're doing work that then gets generalized to other parts of the other areas of science. Now I'll come back and talk a little bit more about that. And so one of the things that I want to talk about is clinical diagnosis, and we call this artificial intelligence. Why is this a problem? Prob, we're meeting at lunch, and so you asked the question, what problem are we trying to solve? And national academies put out a report on diagnostic errors in 2015. If you work in outpatient medicine, patient mortality, adverse events in hospitals, malpractice, diagnostic errors are a big problem in medical care, even in the 21st century. And one thing I always wondered is this especially significant in ophthalmology? Because a lot of what we do in my little piece of the world is we look at the structure of the eye, and could that be subjective and descriptive and qualitative? And so anyway, I'll talk a little bit about that. One of the diseases that I have studied in my academic career is a disease called retinopathy of prematurity. Stevie Wonder was someone who went blind from ROP. And it's a disease that I grew up studying with Paul back when we were in New York City together. He was at Cornell. Actually, both he and Mark were at Cornell. I was at Columbia University. And so the way that this disease was first described in the 1940s, because before then, premature babies didn't survive long enough to get eye disease. And the way that you diagnosed it back then was that you looked at the structure of the retina and you used words and pictures to describe what you saw. Okay, so classic old school ophthalmoscopic diagnosis. Okay, so purely subjective and qualitative. In the 1980s, that changed when there was an international classification system that was developed for this disease. And it happens that those terms and the parameters in that system are things like zone, stage, and something called plus disease. And when everyone in the world uses the exact same terms to describe what they see, there's huge benefits for clinical care. And it also gives you an infrastructure to do multi-center clinical trials. And I would point out that even in 2022, there's a lot of fields in medicine that don't have that standardized terminology. Okay, so when you do have that standardized terminology, you do those clinical trials, and of all these different parameters, it turns out something called plus disease is the most critical one for identifying when a baby's going to go blind and when you have to treat that baby. Okay, it happens you treat the baby with laser. Okay, when most of this work was being done. So what's plus disease? It means that you've got tortuous and dilated retinal vessels. Okay, so that seems simple enough. Okay, tortuous and dilated retinal vessels, and tens of millions of dollars of NIH money went into proving that this is the critical thing and that we have to treat it. Okay, so toward the beginning of my career, this is 2007, we did what I thought was an embarrassingly basic study where we put images up onto the web and we asked 20-some world experts, look at the image, is it plus or is it not plus? Okay, now it happens there's an intermediate category, pre-plus. But in this example, 50% called it plus, 50% called it not plus. We had examples that split 60-40 and 70-30. And this is a big problem. If this is what distinguishes if babies are going to go blind and you have to treat it, yet the world experts are splitting a 50-50. And it happens that if you get a lot of experts to look at a lot of different images, there's some really interesting stuff that happens. And the two people who really did the work were Pete Campbell, who's a retina doctor in Oregon, and Jayshree Kalpathy-Kramer, who's a longtime collaborator from Boston, and actually now in Colorado. So these 8 experts, the way to read this, they looked at 100 images. If that expert diagnosed that image as plus disease, the box is in red. If they diagnose pre-plus, that intermediate, the box is yellow. If they diagnose normal, the box is in green. Okay, and so the first thing to notice is that experts 7 and 8 here at the bottom diagnose plus disease 6 times more often than expert number 1 at the top. Okay. If you go to a different data set here, 34 different images, it's that exact 6-to-1 ratio. Okay. And in fact, people will say, well, obviously we've got the wrong 8 experts to be so variable. The most recent iteration, like there's only an iteration of this international classification system every 15 years or so. The most recent iteration was last year. It was published, and we did this. Paul and I and others did this with the 30-some experts in that international consensus committee. It was that exact 6-to-1 ratio. Okay, so this is a huge problem in clinical medicine, and I don't think it's just an RLP problem or an ophthalmology problem. I think we see this. Those of you who take care of patients know that there are just some doctors that are aggressive, and there are some that are conservative, and this is what it looks like on a grid. Now, we'll look at it another way. If you go down the columns here, so each image is a column, right? And if you calculate a score, give it 1 point for normal, 2 points for pre-plus, and 3 points for plus. And if you average it all out and convert it to a color, that's what it looks like here, okay, the average. And on the left side, the images are very red because everyone diagnosed plus, okay, severe. On the right side, the images are really green because everyone diagnosed a pre-plus and everyone diagnosed normal. But in the middle, there's every shade in between. Okay, and so the point that I'm trying to make with this is that the real world is a continuous spectrum of abnormality. And when we talk about evidence-based practice, it implies that patients come in buckets, right? Normal, mild, moderate, severe. But people don't come in buckets. They come on a spectrum. And I think it's our job as clinicians to put people into buckets, for the purposes of evidence-based practice. But the problem is that some people draw the line here, some people draw the line here. And intuitively, that's one of the reasons that artificial intelligence can help. Okay. Now, I want to just talk about here classifications versus comparisons. Now, you can ask these questions about diagnosis 2 different ways. One of them is, what's your diagnosis? Plus, pre-plus, or normal. That's a classification diagnosis. Any field of medicine, mild, moderate, severe. And we don't agree very well. Okay, those 8 experts on the previous slides, and these are weighted kappas. How well do 2 experts agree in diagnosis? The darker, the better. And this is the basis why the reds, yellows, and greens look so different. If you ask the question a different way, show them 2 images. Which one's more severe, number 1 or number 2? We agree really well on relative severity. Okay, and so in a way, it's not a shock that ophthalmologists are really good at number 1 or number 2, which is worse. But I think that's something that we're going to come back and talk about. But in fact, if you ask this question, 5 different experts, and ask them compare 1 versus 2, 1 versus 3, 1 versus 2 versus 3, 2 versus 4, and just do that thousands of times, you can rank order 100 images from least severe on the average to most severe on the average. And people agree really, really closely on that. Okay, we use these ELO methodologies, okay, the way that people rank chess players in tournaments. And so who did this work? Adam Hanif, Pete, and Jayshree. So I'm going to come back and talk about that. that we're really much better at relative severity comparisons compared to classifications. And I've been wondering, does that have implications for how we train deep learning systems? So I want to here move into disagreement in process. Now, here, this really gets into what I'll call the science and the art of medicine. When we were, here, Paul mentioned a guy that we've both worked with in Los Angeles, Tom Lee, and I was on the podium with Tom Lee about 10 or 11 years ago, and we were talking about plus disease, at a national meeting. And so Tom made the analogy of a US Supreme Court Justice Potter Stewart. In the 1960s, they're arguing a court case on pornography. And so Potter Stewart says, I can't define it, but I know it when I see it because it just looks bad. Okay. And he said, well, plus disease is like pornography. You know it when you see it because it just looks bad. And that comment really bugged me for months because I thought, well, we're trying to be scientific about it. How can you just say things just look bad? But on the other hand, anybody who's a clinician knows that sometimes things just look bad. I know that there's a lot of beautiful corneal work going on here. Sometimes that corneal ulcer, sometimes it just looks bad. And the ICU doctors will say that sometimes that patient just looks sick in a way that they can't necessarily articulate. Okay, so I think this is a real phenomenon. And so we got interested in this question. Could variability in the diagnostic process, in other words, if different experts have different concepts of what it means to just look bad, could that explain the variability that we're seeing here, like, the reds, yellows, and greens? And so we thought, well, let's design up a different type of experiment where we'll capture and encode the detailed qualitative thoughts of the 7 most seasoned ROP experts in the world that we could find. Because these were people who had originally come up with the definition of plus disease or the original classification system. And so we spent months sort of selecting images, questions, and we asked them, let's videotape you while you go through and tell us what about those images worries you or reassures you. Okay, and so Nina Hewing, who was a visiting fellow from Germany, and Paul did a lot of work on this. And we basically had a cognitive psychologist analyze hours of videotaped transcript to model their thought process. And so here's an example. There's an image, and one expert diagnosed plus, one expert diagnosed pre-plus, and one diagnosed normal. And they're all looking at different parts of the retina. Okay, and so I really think there's difference in diagnostic process as well as the overall diagnosis. And so that's what I'll call like science and art of medicine. And we got intuitively, I think every clinician probably knows that different doctors look at different things when they make up their assessments. And we looked in the ophthalmology literature, and it happens that there was another ophthalmologist in flyover country, Paul Littker, amazing work at the University of Michigan. He was chair there for decades. In 1976, he worked on a paper that was called Variability of Expert Observers in Evaluating the Optic Disc. That's how you diagnose glaucoma. And he says here, a significant amount of variability exists between observers in designating cup to disc ratios. Furthermore, different methods are used for evaluating the size of the cup. And he goes through and describes the methods. And I thought, wow, we thought we did some cool work in 2013, and somebody did this work like 40 years before that, and it was basically the same rationale. And so I do think that there's really, really good basis that this is not just a ROP problem. We see this in medicine. So that just really takes us, artificial intelligence for ophthalmology. There's been a huge amount of work in AI in the vision field, and I can't resist. And I'm sorry for being so self-serving here. I captured a tweet from Eric Topol about 2 years ago. Of all the medical specialties, most people think radiology is leading the AI movement, but it's actually ophthalmology so far, because there's so much image data combined with annotating clinical data. And I know here there have been people. Paul's done some beautiful work in AI. Xincheng Yao here, Jenny Lim. There's been a lot of work in this at this institution, and but it happens that the first FDA cleared autonomous AI system in any field of medicine was by a guy, Mike, that you worked with, Michael Abramoff at the University of Iowa. And and the folks at Google have done some beautiful work in knowledge discovery for systemic health. Those of you who are ophthalmologists will know this, and those of you who aren't, they trained AI systems to, based purely on the appearance of a retinal image, identify with almost complete accuracy is that from a male patient or a female patient. Okay, and that was stunning to the community in 4 years ago when it was published, because none of us can look at a retina and tell, is it male or female? But presumably there's some data in pixels that confers sex. And so, obviously, can we come up with ways to take advantage of that from a knowledge discovery perspective? And lastly, in our little part of the world, people like Paul, me, James Brown, Pete, and Jay Cherie have developed systems for plus disease diagnosis and ROP. They beat experts in terms of performance, plus, pre, plus, or normal. Okay. So trying to figure out how are we going to get this into the real world. But anyway, I want to talk about this paradigm going from qualitative to quantitative. This is what we're seeing all the time in every field in medicine. And in ROP, we've talked about AI. There's other areas in ophthalmology where it's optical coherence tomography imaging. That's sort of we do it in every single patient now. In general medicine, it's going to be imaging instead of listening to the nuance of murmurs. But anyway, in the ROP world, we thought, well, if we're arguing that clinical diagnosis is subjective and qualitative, how do we make it more objective and quantitative? And so we used these AI systems and converted their output to a 1 to 9 scale. 1 is most mild, 9 is most severe. And how can we want to do that? From a clinical perspective, what you want to know as a doctor is my patient getting better, worse, or the same compared to my previous exam? And our current scales are 3 levels, right? Plus, pre, plus, normal, or mild, moderate, severe, whatever your disease is. You can't do that with sufficient granularity on a 3-level scale. So we thought, well, maybe 9 levels can be good for that. From a practical perspective, every time I would give talks, somebody would come up and say, well, it's great that you've got this computer system. Am I at medical legal liability though if the computer system says treat and you don't treat? And so I'm not necessarily saying I agree with that. In fact, I don't. But I've heard it so many times that we thought, well, let's come up with a number, and it's like a piece of data for the clinician, like any other piece of data. Doctors don't, blood pressure cuffs don't diagnose hypertension. Doctors diagnose hypertension based on data that comes out of blood pressure cuffs. And so we thought, well, the same thing. This gives data for the clinician to decide. And maybe if you went from a 3 to a 6. That's different from going to a 5 to a 6 in 1 week. And lastly, from an experimental rationale, we talked about comparisons being better than classifications. Right? And well, maybe if we have 9 standard photos, you can take your photo as a doctor and line it up, better or worse, and where does it belong, with in principle really high accuracy. Okay, so that's what we're talking about in this part of the world, but I think this is a generalizable thing across medicine, qualitative to quantitative. Now, I'm gonna skip over this and just say that people always ask, well, garbage in, garbage out. What if you have a poor quality image? A grad student, Aaron Coiner in Oregon, who I worked with, trained deep learning systems with very high accuracy to with human labels, acceptable, possible, acceptable, not acceptable. How do you give a quality score? For example, from 0 to 1. And so, these seem to work really, really well. And we're trying to figure out how to get these into operational AI systems, okay, to pre-screen your images. So I'm going to, wrap this part up and say, what have we learned about this disease? Well, there's science, there's art, and there's technology. And, the science here is classification systems in clinical trials, right? They're really, really amazing because they've revolutionized clinical care, but they're not perfect because, at the end of the day, clinical diagnosis is subjective and qualitative. And that gets into the art where, evidence-based practice cannot capture all that qualitative nuance of disease, right? What does it mean to just look bad? And that takes us into technology And in this case, it's AI, but I would say, we're going from qualitative to quantitative. Okay, so I think that, one of the things I hope everybody here, remembers that whatever, our individual piece of the world is, that good care needs all three: science, technology, and art. It's like being nice, bright, and hardworking. Like you need to have all three. Okay. So with that, where are we heading? At the National Eye Institute, we published a new strategic plan about 9 months ago. So Paul talked about all this gray hair, and some of it is because of this strategic plan that we published 9 months ago. In a nutshell, our portfolio at the Eye Institute is anatomic. Okay, there's retinal disease, corneal disease, glaucoma, and our plan is organized methodologically. Okay, genetics, neuroscience, immunology, regenerative medicine, data science, quality of life, and public health and disparities. And the reason that we did this is that academic eye departments are organized anatomically. There's a division of retina, for example. Basic science departments are organized methodologically. And so we thought, well, this would be the best way to try to match the methodology with the application. Okay, so I'm going to talk about data science because it's relevant to AI. And 21st century science, we have unprecedented access to large-scale data. And it happens that regardless of whether you're looking at biological data, omics data, imaging data, clinical data from EHRs, or population-based data, it's this core set of methodologies. And I do think that we're fortunate in vision science to have data types very easily accessible along that full spectrum, from biological data to population-based data. And so if AI is so good, why aren't we all doing it now? Just some very quick unanswered questions. Number one, these AI systems are really good for diagnosing very, very narrow tasks. Do you have plus disease in ROP, for example? The real world, has a lot of parallel tasks. Doctor looks at a patient, they have a lot of different findings. And, it's not just about looking at pictures, it's also about integrating that with clinical data. So I think we've got a lot of synthesizing to do with that. Number two is what I'd call generalizability and bias. And during the past day and meeting with several of the faculty, this has come up a lot, where a lot of AI data comes from very homogeneous populations. I used to work in Oregon, and the real world is heterogeneous. Okay, there's different imaging machines, there's different races and different populations, and in general, you cannot take a study validated in one thing and then move it to a different imaging device, different race, different population. And so this is a huge problem in this country and around the world. And the other issue is that external validation, only a study in the Korean radiology literature, only 6% of deep learning studies had any external validation. And so you all know that during COVID, AI was supposed to solve all of our problems, and it really didn't. Okay. And so lastly, we've touched on this unclear ground truth. Okay, sometimes there's variability among raters. Okay, sometimes the difference between mild and moderate really isn't that clear. And often we have very unclear definitions of ascertaining endpoints. Okay, how do you define moderate disease? If your disease happens to be glaucoma, how do you define rapidly progressive glaucoma? It gets down to that issue of difference in process. Okay. So there's a lot of work, and this is just a snippet of what's missing. So what do we need? This is one of these. I added a couple of slides, last night after dinner, and in between, based on things that we talked about. One of them is that I think that 21st century research relies on big data, right? And we really need better mechanisms of data sharing and standard data representation. Okay, we've got this paradigm in medicine and science where there's often multiple, what I'll call small studies all around the country that are kind of trying to answer the same question. Okay, but individually, they're all underpowered to truly answer that question and advance the field. And oftentimes these investigators are not incentivized or even feel disincentivized to share their data, okay, like we talked about. Now, the bottom line though is that even if investigators did want to share, you generally can't, because when people collect their data differently, you can't just harmonize it. So So data sharing and data representations. Now what are we trying to do about this? At the NIH, we have a data sharing policy that goes into effect next year. And the White House has guidance, a policy statement about this as well. And those are sticks. Okay, and I really think that we need carrots to truly incentivize data sharing. And about a year ago, we worked on one little project. There's a guy, Marco Zarbin, who's the editor-in-chief of a journal called TVST. It's the open access journal of one of the major research societies in ophthalmology, of the major research society. And Pierce Keane and Aaron Lee are two of the editorial board members for TVST. And we basically defined, I actually got all the editors-in-chief of the major vision journals together. And we thought, well, let's try to create a new article type in vision science called a data set or software library. And this was the first journal to instantiate that. Now there's a second journal called Ophthalmology Science. But we thought, well, people can maybe get academic credit. You can cite your data, find it on PubMed, and maybe that would be one way. But we need a lot of other ways to truly incentivize data sharing and team science. Okay. And I do think that we need a common data model for eye care, along the line to solve this problem of inconsistent data representation. And one of the things that we're starting to do at the Eye Institute is to collaborate with the American Academy of Ophthalmology to develop that common data model. And there's a person, Sally Baxter at UC San Diego, Carrie Getz at the National Eye Institute, and Michelle Rybar in Oregon, who's taking a sabbatical at the NEI to lead this effort. Okay, so I hope we're moving forward. Another thing that we need is imaging standards. And this is a huge problem across all of science and medicine, where in the vision world, a lot of data comes out of machines, right? They generate images and they generate numbers. But the problem is that these data are often locked in proprietary standards where you really can't get access to those data. And these data, as shown before, are the whole basis for data science and artificial intelligence. And this is a huge problem from a clinical perspective and also from a research perspective. The department that I used to work in maintained two image management systems because the devices and the data management systems couldn't talk to each other. Okay. And so in May 2022, we convened a workshop that was collaborative with 3 federal agencies. And so we actually all got together and we planned this for like 9 months. Us, and because we fund, our thing is that we fund research projects, right? The FDA, that was led by Malvina Edelman here, because they regulate devices. And the Office of the National Coordinator for Healthcare IT, led by Mickey Tripathi, because they regulate electronic data exchange. Okay? And so we thought, well, what can we do together from a federal government perspective to try to promote interoperability? And in follow-up to this, the American Academy of Ophthalmology and ARVO, which is the major clinical society and the major research society, put down an editorial about this. And we're working on our follow-up. What are we going to do? But I've really been heartened that the vendor community seems to have responded, very, very well to try to work, with us on this. Okay, so this has taken a long time. I've struggled with this for 15 years. I think we need larger data sets that are AI ready and that avoid bias. And Bridge to AI is a NIH Common Fund project, trans NIH, basically building large data sets. So this is different for NIH. We're traditionally hypothesis driven. This is hypothesis generating. Okay, and I got the number wrong here. It's actually $130 million over 4 years for data generation, and a different paradigm. And I think that a few weeks ago I was really excited that one of the 4 groups that got awarded these data generation projects was actually led by a group of ophthalmologists. Okay, Aaron Lee and Cecilia Lee at the University of Washington. Okay, but a lot of work that's can be done. I think this is going to be exciting. Now, Aim Ahead is another trans NIH project that you should know about. Aim Ahead is a project that's led through the Office of Data Science Strategy. Okay, that's led by Susan Gregorek. And a couple, about a year ago, the question was, how can we spend $100 million on some topic involving artificial intelligence, machine learning, and EHRs? And this is the topic that we chose. AI to address challenges of health disparities and minority health. I mentioned that here because there is such a rich infrastructure that I can sense here in terms of working on health disparities. I know that, I'm looking here at Charlotte, Jocelyn. I know Angel Scanzera is doing some beautiful work in this area. As our a few other Naranya, I know is out here somewhere. And so what does this mean? It means developing diverse data sets, right, that represent the whole population, training a diverse workforce. Okay, some of these community health clinics, really actually don't have access to EHRs, let alone machine learning, but how do we get them up? And lastly, how do we detect AI approaches to avoid bias and to detect bias? Because what we don't want is algorithms that have bias built in. And one of the things is that I think that a lot of the sources of bias in the EHR come from subjective observations that clinicians write into the chart, like the patient was non-compliant, and a million other things that people can write. And so maybe I think we need objective image data, to help anchor that a bias and to really try to study, well, what is the source of this bias and how do we avoid it? Okay, so if you're interested in this, just look into that, because I think this is a great institution. I think we need a better medical education as pertains to data science. I would argue that the one tool that every clinician and scientist in this country uses is not a stethoscope. It's not an ophthalmoscope. It is a computer. Okay, and despite that, there is basically no systematic training or framework about how to use those technologies. Okay, people learn by trial and error. Okay, and so compare that to if you're a doctor, learning how to take a history, do a physical exam, listen to the heart. Medical students will spend months to years of dedicated time learning how to do that. But the amount of time you spend with learning about understanding technology is generally zero in medical schools. It's just trial and error. And furthermore, the paradigm that we have in medicine is that the person with less gray hair learns from the person with more gray hair. And in this specific case, it's usually the person with more gray hair who knows less than the younger person. So we really have to figure out how to turn that paradigm upside down. And I just want to highlight that there were a couple trainees here at UIC, Emily Cole, Nita Velikadath, and working with Paul Chan and some others who wrote a really, really nice editorial about this, that's ophthalmology specific. Okay, but I think this is a very, very broad problem. Now, I think another thing that I specifically want to talk about is that I think we need methodological innovation. And I just want to give an example here. Retinal prosthetic devices are one, for patients who really can't see well. When I was growing up, we could give them a magnifier, or a computer would read the screen instead of having the patient read it. Retinal prosthetics are an emerging approach to that. But the example here is that this is work that was done by Daniel Polanco at Stanford, together with Jose Elaine Sahel, who was at University of Pittsburgh. And the patient wears this device. It has a camera built in, parses the surroundings, sends signals back to the retina with infrared pulse of light. And there's a subretinal implant here that gets put in that then sends signals to retinal cells to stimulate the retina to give the sense of vision. Actually, really, really remarkable results here. But what I found fascinating is that what really went into this was advances in semiconductor physics, wearable electronics, neural engineering, visual psychophysics. In other words, there were Stanford PhD students who were writing their theses on the technology that went into this device. And there were Stanford junior faculty that were getting tenured for working on those projects. And what we really want to do is get the best methodologists focused on solving innovative problems, as opposed to taking off-the-shelf methodologies and applying them into the eye. I thought this was a beautiful example of that. And the reason that we had a really interesting dinner conversation about this last night with Prob. Tom Royston from biomedical engineering was, we were sitting there and just talking about this. And one thing that really impresses me and excites me about this institution is that I think that you have a very collaborative atmosphere and an atmosphere where you have really, really good methodologists with really, really good clinicians. And I hope that's an area where we can really make this stuff happen here. And lastly, in terms of delivering access to care, this pandemic really exposed a lot of the disparities in this country that you all at UIC, I think, have known for a long time, because I think it's what you've been doing. But how are we gonna address that in vision care? Maybe it's going to be through telehealth. Maybe but if we do that, we're going to have to evaluate how to implement these systems. Do they work? Are they cost effective? And I think we're going to have to develop new models for remote care that are not going to look just like, well, let's create a CPT code, for what it is. Someone's got to create that new model and validate it. I think we're really going to need to understand what are the social determinants that are relevant to vision health. In other words, why are people going blind from treatable disease even in 2022? So I think a lot of work here. And lastly, what's the impact of technology on practice? Anybody knows that EHR notes are getting longer and longer than ever before. How come? People are copy pasting. They've got buttons that say everything is normal. And from a visceral perspective, did those things really happen that day? Nobody really knows. There's a diagram like this that represents serum chemistries. It's been 25 years since I worked up an internal medicine patient, but I can still look at that visual representation and get a good sense of what's going on, because that's that mental model. These days in EHRs, we don't have mental models. We have alphabetized. In some of these things they alphabetize. In some of them they list NAKCL. And so what are the implications of that? I don't think we know. And a couple of years ago, I worked with Adam Rule and Michelle Rybar looking at electronic health record documentation patterns in Oregon. There's almost 3 million notes across 40 different specialties. We wrote little Python script. How long is the note? Where did the text come from? Did someone copy paste it? Did someone type it in? Did it come from a template? And how similar was it to the previous note from the same patient? We wrote these sequence alignment algorithms. And it turns out that the notes are really long. Okay, in ophthalmology, they were 6 pages on the average. The content was heavily redundant. In ophthalmology, 75% word for word, the same as from one to the next. And it's getting worse over time. And so obviously, the questions here, are we doing this for clinical care or are we doing it for billing? And what's the research value of these notes? If this is going to be the substrate of EHR, we've got to, of AI and machine learning, we've got to get it right on the inside. Okay, so anyway, I'm going to finish up just with one quick anecdote. People talk about, well, I hate the new technology. It's like a 8-page note instead of a 1-page note. It's all copy pasted. But there was someone I used to work with in Colombia named Vimla Patel. She was a cognitive psychologist who became interested in informatics. And there was a paper, so this is all Vimla's work here. The scenario is that she worked with a primary care clinic in Canada in about 2000. And so part one of the 3-part experiment, part one, Vimla Patel goes to the clinic, gives the doctor a piece of paper and says, work up the patient and write me a note. Okay, and so here's what it looked like. Okay, 74-year-old woman, diagnosis made in February. She had polyuria and nocturia. She had an eye exam, normal retina. And for anybody here who's a clinician knows that this is what you learned in school, right? We all learned you talk to a patient and you tell a story about what happened, right? This is narrative text. Now, part 2 of the experiment is that clinic converted to electronic health records. And so this is the 2000 version of the EHR, in boxes to check, pull downs, little drawing template. It actually looks like the 2022 version of the EHR. Hey, but part 2 is basically work up a patient with the EHR and write me a note. And this is what it looked like. Okay, completely different. This is structured text. And this is the world that we live in now. Okay, my patient had pain. It was severe, mild, moderate. It was dull, aching, sharp. And so, we've gone from narrative text to structured documentation. Okay, this is a really, really big paradigm shift. Okay, and so part 3 of the experiment is that the clinic is now used to EHR. Vimla Patel goes back, gives the doctor a piece of paper. work up a patient and write me a note. And here's what the paper transcription looked like. Okay. And I thought this was fascinating that it's not just about that 8-page versus 1-page note. It's the tools that we use and that we give clinicians to do their work affects the way that they think about information. And I think this, as a medical community, I think gives us all something to think about, like, how are we going to address these problems, and how are we going to really invent what the future is that blends science, technology, and art, right? And my last slide. I think I've really tried to argue that science, technology, and computing have transformed eye practice. And clearly there's a analogy to medical practice overall. We're going from qualitative to quantitative. Okay, now, if you're doing artificial intelligence, if you're doing OCT imaging, which is the bread and butter of ophthalmology, retina practice now, if you're doing evidence-based practice, I've been wondering about this question of what is your added value? Like, back when I was training, I had to learn to use this device. It's a contact lens that you put up to somebody's eye to look at the retina. And I had to learn to recognize all that nuance of the different parts of the retina. And that was kind of my added value, that I knew how to do that and other people didn't, right? And so 2 years ago, I was back in Oregon, and there was resident research day. So there's 6 residents standing up on stage after they given presentations. 3 of them had worked with me or Pete Campbell on AI for ROP. The other 3 had worked with David Huang and Yali Jia doing OCT imaging, a world-class group that was doing imaging. And so I asked them the question that you're all working on these technologies that go from qualitative to quantitative. If everybody now sees the exact same numbers, okay, regardless of whether you're a retina subspecialist versus a general ophthalmologist versus an optometrist versus a patient using Google search, whoever, like, we're all seeing the same numbers. Like, what's your added value going to be? Okay, and of the residents, not a single one could answer the question. And actually, not a single one looked like they'd ever thought about that question before. Okay, and that really, I think, is a little bit troubling, because as a community, we have to figure out, as we go from qualitative to quantitative, what is our added value going to be? And maybe it's going to be diagnosis versus management. When we diagnose, AI systems are really promising, but you need clinical judgment to know what data to collect, right? If you manage data, that's a different story. Patient has cancer. Are we going to are you going to get chemotherapy and have very, very violent side effects, but survive on the average X number longer? Or are you going to go into hospice and have comfortable time with your friends and your family? Those are human decisions that AI systems don't make. And maybe it's going to be the role of clinicians to help guide patients through those discussions. But if the impact of EHRs is to have doctors copy pasting and not really talking to patients, are we losing those skills that are going to one day separate us from those machines? And so I really hope that we can advance, sort of the science and understand how to use these technologies, and also figure out how we're going to maintain those uniquely human skills. And I think that's going to be the way to sort of combine the science and the art and the technology. So with that, I just want to thank you. What I have tried to do here is to integrate these concepts of what we've learned in ophthalmology together with what we can apply to medicine and science overall. And I really hope that does justice to what the premise of this Dean's lecture intends to do. And just thank you very much for the privilege of letting me speak here today.
Do we have any questions? Yeah, that's right. That's knowledge. Let me.
I really enjoyed your talk, its thoughtfulness and the grand vision that you presented. So I have one very general question. This approach may generate a lot of very interesting questions, a lot of very interesting hypotheses, but not every question is interesting and not every hypothesis is important. So how would one sort of develop an approach that says this is more important to study versus something else?
let me try to paraphrase what I think you're asking, because I think it's a really important question. With more and more data, there's more and more potential questions out there, and how are we going to know what to do? I view this as sort of a two-pronged thing. One of them is that, of course, I have faith in the ability of individual people who are really smart and creative to identify questions to ask on their own. And I hope that the community will help to, police itself in the sense of, gosh, this is a really neat thing. This is important to do. And, but part two of that is that I see that as part of my job at the NIH to try to identify, okay, what are some things that we think, because presumably we can see the breadth of the field in a way that, individual people, may not be able to. And so I hope that with a combination of, people in the real world coming up with individual questions versus us, from a bird's eye view, identifying, well, what do we think are some of the important questions? I hope it's going to be that combination that gets at what you're, really saying. But, if other of you, I think we are entering a new world where there's more and more stuff out there than ever before. And if any of you have ideas, please, that, that's why I put things like social media up there, that, I really want to hear it. But thank you.
Mike, great presentation. And I think your talk really, I think, convinced me that for a long time, researchers in medicine have driven therapeutics and driven our approaches in some ways. And, we've pushed ourselves into evidence-based medicine, which is good. But I think one of the things that I sense, especially in your example, retinopathy, is actually how important clinicians are going to be in the next era of data science. And I say that because it seems to me that the categories that we use in our diagnoses are so much more important given the power of data science. And so we need to have accurate categories and categories that are clinically grounded, not theoretical, in order to drive the data science forward. And the clinicians are the ones who sit in that space more than us who are researchers in some ways.
Yeah, Niranjan, a couple people told me how smart you are. And I think you just articulated something that took me years to really think through. And if I can try to elaborate a little bit, or maybe even repeat what you said in sort of clinician terms, one thing that has really struck me is that the premise of evidence-based practice is that there are these categories that are deemed to be what's mild, what's moderate, how do you define severe? And how do these categories get there? Because experts, somewhat arbitrarily, somewhat based on expert consensus, defined them 40 years ago, or however long it was. Somebody did a clinical trial and validated that it works. And then now we teach it to each other, for decades, and this gets into the vernacular. And now, in fact, in my little piece of the world, ROP, we just showed that the basis for one of those categories, people can't get accurate, plus disease. But I think there's a deeper part of that, which is, how do we know that those categories are really the right way to define those cut points? And can we use technology to maybe even from first principles come up with a categorization system that makes more sense? I actually think that would be fascinating, and it would require sort of scientists and technologists and clinicians to work together really, really carefully to try to redefine what those things are. But I actually would be fascinated by studies that do that, because I don't think they're easy to do, but I think that they could really advance the way that we think about disease and like sort of science and disease.
Any other question?
So thank you again for introducing us to the wonderful application of AI in ophthalmology. And one of the questions I have is, you showed how nicely classification is essential for using AI, but the added value in the human skills is probably humans building a causal narrative and building a causal model. And I think what you saw in how EHR transformed how we write notes was that the initial narrative used to have a causal model. There was an idea that this is the pathogenic mediator, this is where the patient is heading. Do you see a future for building more causal models in AI, or do you think that AI's primary role in the next decade will be to do accurate classifications, and it is the humans who will need to be trained to use that data to build the causal pathogenic models?
That's a great question. And so the answer, of course, is that I don't know, but I'll give you my opinions about this. I think that I would love if these computer systems can give us insight on, for example, what does it mean to just look bad? And I actually truly can see that around the corner. And we'd have to think carefully about how to design that. There's a lot of circularity in this, but from another perspective, you can look at causal model a different way, sort of mechanistics of disease. Like, yesterday and today we talked with like Andreas, who is looking at sort of mechanistics of diabetic retinopathy. And maybe there's going to be a way to look at causal model that integrates clinical imaging together with mechanistics of disease from a pathway level. That would be another way to look at this. And so I think, in a nutshell, I think causality and collaborative work could span science and clinical in all sorts of interesting ways. So I just really like that. I actually love that premise.
The last question, please.
Hi, Dr. Chang. Thank you for the great lecture. I'm a young scientist trying to make new technology and disseminate that to other clinicians. But personally, I've had the challenges of trying to convince clinicians to adopt new technology into their practice. And I was wondering if you had any advice or anything that NIH is doing to actually convince established clinicians to adopt new technology into the practice. Thank
you. That's a great question. And I don't think there's an easy answer to this, except to say that, so there's facts and opinions, and I'll give my opinion about this. That I, on the first day of medical school, every student sits in an auditorium like this, and they're told that half of what you learn in the next 4 years is going to be obsolete, right? And I never believed it, but it's totally true. Okay. And in the field of medical retina, 100% of what I learned virtually is obsolete in 2001. And my opinion about this is that we have to teach medical students the skill set that's going to help them be the best doctor in 2040, not the best doctor in 2022. And I mean, to be perfectly honest, sometimes I see medical education moving more and more toward, let's teach you the body of knowledge that you need to pass your board exams in 2022. Okay, and so I think that part of that problem is going to be making sure that we train a group of clinicians that's, number one, interested in how am I going to advance my clinical practice? And just because there's known and there's unknown, and you want to take known and make it bigger. It's not a constant. Because I honestly do think that in clinical medicine, that's a mindset that we have sometimes. And I think it does boil down somewhat to training. And I think that from a collaborative research perspective, I think the more that we can have clinicians get used to the fact that, well, it is part of your job to take the known and expand it. And it's part of your job to work with the researcher to develop those new, whatever, the technology, the science, the new small molecule, to help you do your job better. I actually think our whole community benefits from that. But I view that as a medical education, kind of issue. It's not easy, but that's my candid opinion.
Thank you. Wonderful lecture and faculty under my account downstairs. Please join us there. You can continue conversation with the speaker as well as thank you all very much.
Can you